Hello, my name is Faith Holloway, and I'm a senior biological sciences major at Chapman University. I'm here to discuss today a study that focused on a new method of detecting the presence of pancreatic ductal adenocarcinoma, a particularly deadly cancer of the pancreas. Today, pancreatic cancer remains to have one of the lowest 5-year survival rates of all cancer types at only 9%. This is primarily because pancreatic cancer is very difficult to detect in early stages before the cancer has spread to other organs. Patients typically do not begin demonstrating symptoms of pancreatic cancer until the disease moves beyond its early stages, at which point it is much more difficult to treat. Furthermore, pancreatic cancer diagnosis can often be an invasive process requiring the removal and analysis of tissue samples from the pancreas. Because of this, screening for pancreatic cancer is not particularly widespread. Physicians and researchers are constantly on the search for new ways that are non-invasive and highly sensitive to detect pancreatic cancer at early stages when it is most treatable. A scientific paper published in 2016 by UCLA researchers considers the usefulness of circulating tumor cells as a method of diagnosing pancreatic ductal adenocarcinoma. A tumor is essentially a mass of tissue consisting of abnormal cells that are growing uncontrollably. Cells are the basic building blocks of every tissue in the human body. Each cell contains a copy of genetic material, or DNA, that is essentially a blueprint for life. The DNA contains instructions for making proteins, which are large, complex molecules that carry out metabolic processes and are responsible for keeping us alive. In cancer cells, something in the DNA has been altered. This mistake in the DNA is referred to as a mutation and results in the production of an abnormal protein that does not do its job correctly. One mutation that is highly correlated to pancreatic cancer incidence occurs in a region of the DNA corresponding to a protein called KRAS, which functions as a molecular switch for cell growth and division. When this protein is mutated, it can no longer effectively control cell growth, and cells become capable of dividing uncontrollably and forming a cancerous tumor. In this study, researchers attempted to match KRAS mutations from primary tumors of the pancreas to KRAS mutations in circulating tumor cells using a pool of patients diagnosed with pancreatic ductal adenocarcinoma. Circulating tumor cells are cells that have been shed off of a primary tumor and into the bloodstream or lymphatic system. Because circulating tumor cells are present in the blood, they provide a much less invasive alternative to diagnosing pancreatic cancer, as screening for mutations could be done using blood samples rather than tissue samples from the primary tumor. I will now be discussing Figure 3 in the research article titled Circulating Tumor Cells as a Biomarker for Diagnostics and Staging in Pancreatic Cancer. The scientists first obtained blood samples from each pancreatic cancer patient used in the study. These blood samples were processed to isolate healthy white blood cells and single circulating tumor cells. Tissue samples from the primary tumors of the pancreas were also obtained for each patient. DNA was isolated and amplified using modern molecular biology techniques from circulating tumor cells, white blood cells, and primary tumor tissue from each patient. A procedure was then used to amplify the region of the DNA corresponding to the KRAS protein. The researchers then analyzed the amplified KRAS region in each sample to determine what had gone wrong in the blueprint to cause cancer. They discovered that white blood cells from the patients had normal DNA and produced normal KRAS proteins. The DNA from circulating tumor cells and primary tumors, however, was slightly altered at a specific location within the KRAS region for each cancer patient. The error in the DNA from circulating tumor cells and primary tumors have resulted in a non-functional KRAS protein being made. This error occurred at the same position in the DNA for all patients. Furthermore, the DNA errors found in circulating tumor cells matched those of the primary tumor for each patient, confirming that the circulating tumor cells had in fact been shed from tumors of the pancreas. Thus, these circulating tumor cells could be used to indicate the presence of pancreatic cancer and a much less invasive diagnostic method than tissue extraction from a primary tumor. If pancreatic cancer could be detected using circulating tumor cells found in blood samples, patients could be more proactive about screening, and more cancer cases could be detected in early stages when they are still fairly treatable. Thank you.